dear students, welcome to lecture series in Botany. Today we are going to discuss a very important topic, which is molecular taxonomy. Taxonomy is the discipline of biology that deals with description, identification, naming and classification of organisms. It's the very foundation of biological sciences. Traditional approach to the measurement of biological diversity rely upon the ability to resolve differences in morphological characters. Fieldwork, collections, diversity surveys, floras, monographs and conservation efforts provide the primary means for working with physical materials needed for investigations in systematics and genomics, as well as for formulating hypotheses, interpreting the results and making useful applications of those results. In last more than half a century, the advent of molecular biology has revolutionized the entire realm of biological science. Molecular biology provided us new tools to decipher genetic information. Molecular biological techniques have diverse applications, from early detection of abnormalities in the embryo and saving prospective parents as well as the child from ordeal to targeted delivery of drugs to the diseased tissues, in solving disputed paternity cases and in aiding criminal identification. This new science has influenced the field of taxonomy immensely. The use of molecular biology techniques in attempts to reconstruct the evolution of organisms and to evaluate and revise existing classifications has given birth to a new field of taxonomy known as molecular taxonomy. That is precisely what we are going to discuss. Molecular taxonomy is defined as the use of molecular data, especially from DNA, in classifying organisms. Now the question arises as to what is the source of molecular characters. Well, there are three basic sources, that is genomes in eukaryotic plants, from which molecular data can be acquired. These include chloroplast DNA, mitochondrial DNA and nuclear DNA. In addition to these, RNA is also used in taxonomic studies. Genome mapping also provides information relevant to plant taxonomy. While chloroplast DNA is 135 to 160 kilobase pairs, mitochondrial DNA is much variable in size, ranging from 200 to 2500 kilobase pairs. Both these organelles replicate independent of nucleus, have circular DNA and uniparental inheritance. Nuclear DNA varies in length from 1.1 into 10 raised to power 6 to 110 into 10 raised to power 9 kilobase pairs. Nuclear DNA is inherited biparentally in sexually reproducing species and in some model plants has been sequenced completely. Chloroplast DNA has found wider applications in molecular taxonomic studies. The RBCL gene, which encodes the large subunit of ribulose 1,5-bisphosphate carboxylase oxygenase, that's Rubisco, a very vital enzyme in photosynthesis, has been used extensively in molecular taxonomic studies. The study of nuclear DNA has traditionally involved ribosomal DNA because it has high copy number. So, this was about the source of molecular characters. Now, the techniques which make use of these sources. First technique is the restriction fragment length polymorphism. It's abbreviated as RFLP. It's pronounced usually as RFLP. It involves incubating DNA samples with restriction enzymes, what is commonly known as restriction digestion of DNA. The DNA fragments are then subject to electrophoretic migration in an agarose gel. Fragment pattern on the gel is preserved by southern blotting, whereby DNA fragments are transferred from the gel onto a nitrocellulose membrane. The DNA bound to nitrocellulose membrane is hybridized with a labeled DNA probe, washed to remove any unhybridized probe and photographed. An RFLP occurs when length of a detected fragment varies between individuals. RFLPs are codominant markers and enable distinguishing heterozygotes. RFLPs give highly reproducible patterns, but the technique is not without faults and has following limitations. RFLP analysis requires a large quantity of relatively pure DNA. It's a laborious technique. It requires a good supply of probes. The blotting and hybridization steps are time-consuming and are difficult to automate, and choice of restriction enzymes influences the results. Now, the second technique is random amplified polymorphic DNA, abbreviated as RAPDs, RAPD. This technique involves random amplification of DNA from a sample using an oligonucleotide primer pair. The amplification reaction is carried out at low annealing temperatures. The primers bind several sites on complementary sequences in template genomic DNA and produce discrete DNA products. The amplification product is subject to gel electrophoresis. Nucleotide variation between different sets of template DNAs result in presence or absence of bands because of changes in the priming sites. The bands are visualized under UV transillumination after proper staining. RAPD markers are dominant and do not clarify whether DNA segment is amplified from a heterozygous or a homozygous locus. RAPD requires a very small amount of DNA, involves no blotting or hybridizing steps, therefore it's quick, simple and automatable. However, it cannot be used with degraded DNA samples and its resolving power is much lower than targeted species-specific DNA comparison methods. This technique involves PCR amplification of DNA, therefore the quality and concentration of template DNA, concentration of PCR components and the PCR cycling conditions may highly influence the outcome of results. Mismatches between primer and the template may result in total absence of PCR product as well as in decreased amount of the product. Thus, RAPD results can be difficult to interpret. Now, the next technique is amplified restriction fragment length polymorphism. It's abbreviated as AFLP. In this technique, the genomic DNA is digested by two different restriction enzymes, a rare cutter and a frequent cutter. Double-stranded adapters are then ligated to the ends of restriction fragments, which are then amplified by PCR using primers that correspond to the adapter and restriction site sequences. These primers have additional nucleotides at the 3' ends extending into restriction fragments in order to limit the number of fragments that will be amplified. The AFLP products are detected by labeling one of the two primers, and the labeled DNA fragments are separated by electrophoresis in denaturing polyacrylamide gels, similar to sequencing gels. Typically, 50 to 100 amplification products are detected in a single lane. Polymorphic bands can be identified by comparing the amplification products derived from two lines. Like RAPDs, AFLPs are typically dominant markers. The AFLPs appear to be as reproducible as RFLPs, but they are technically more demanding and require DNA higher than that of RAPDs. Now, the next technique is variable number of tandem repeats. It's another powerful technique for studying genetic diversity. VNTR technique utilizes hypervariable regions of a genome comprised of tandemly repeated simple sequences. These sequences vary in number and hence in length and are therefore generally called as VNTRs. Although the term microsatellite or simple sequence repeat, that's SSR, and mini satellites are used where the basic repeat unit is around 2 to 8 base pairs in length or longer respectively. Specific primers are used to amplify a small genomic region, which is 150 to 250 base pairs, that contains a polymorphic microsatellite sequence. The size of amplified fragment will vary depending on the number of repeats present in a given line. These polymorphic fragments can be separated and visualized by electrophoresis in agarose gels or polyacrylamide gels. The advantage is that no restriction endonucleases are used and the problems associated with partial digestion of DNA are avoided. These are codominant markers and produce highly reproducible profiles. However, long time is required to design PCR primers that give good results. Now, the next technique is a single strand conformation polymorphism, SSCP. In this technique, the target sequence in DNA is amplified and simultaneously labeled using radioactively labeled PCR primers or DNTPs. Subsequently, the PCR product is denatured and analyzed by electrophoresis on a non-denaturing polyacrylamide gel. Mutations are detected by altered mobility of separated strands relative to the strands of the normal wild-type PCR products. This approach shows structural alterations including point mutations in PCR products. The use of SSCP systems based on a capillary electrophoresis, that CE, has increased analysis speed and lowered reagent consumption of this technique. However, SSCPs have a variable mutation detection rate. The sensitivity of this method varies between 80 to 95% depending on different conditions of electrophoresis. Now, the next technique is heteroduplex analysis, abbreviated as HA. HA detects base changes in double-stranded DNA subject to electrophoresis in non-denaturing conditions. In this technique, denatured control DNA is mixed with denatured sample DNA. The DNA mixture is then allowed to re-anneal. The heteroduplexes thus formed are analyzed on a gel optimized to resolve conformational differences. Any heteroduplex migrates more slowly through the gel as compared to its homoduplex counterparts. Therefore, any sequence change can be detected by an extra band above the main DNA homoduplex band. Heteroduplexes with mismatches migrate slowly through the gel matrix on account of aberrant distorted structure with bubbles or bulges at the site of mismatched bases. Heteroduplex analysis can be easily applied to large number of samples and is particularly suitable for analysis of PCR products. The mutation detection rate of HA method is about 80%. This method is more suitable for detection of insertions and deletions and has lower sensitivity in detecting base substitutions. Now, the next technique is denaturing gradient gel electrophoresis, DGGE. This technique separates DNA strands on the basis of their actual base composition or the ratio of GC to 80 base pairs. Typically, DNA is amplified by PCR. Amplification is performed with genes or segments of DNA that are common to all the organisms that may be present. The amplification product is subject to gradient of denaturant at elevated temperatures within a polyacrylamide gel. As the DNA progresses through the gel from lower denaturant concentration to a higher one, it starts to melt. Samples with lower GC content melt more rapidly, therefore they progress more slowly within the gel, thus becoming separated from other faster moving strands of DNA. The separated bands can be seen under UV illumination. Now, the next technique is a temperature gradient gel electrophoresis. It's abbreviated as DGGE. This technique works the same way as DGGE. The only difference between the two is that while chemical denaturants, that's urea, and Formamide are used in DGGE. The denaturant used in DGGE is a gradient of temperature, hence this name. Next is DNA barcoding. It's an efficient method for species identification with a great potential to contribute to taxonomic and biodiversity research. In this technique, a short DNA sequence, the barcode, is retrieved from a standardized part of the genome from the specimen. This sequence is compared with a library of reference barcode sequences from individuals of known identity. The specimen is identified if its sequence closely matches one in the barcode library. Otherwise, the new record can lead to a novel barcode sequence for a given species, or it can suggest the existence of a newly encountered species. Subunit 1 of mitochondrial cytochrome oxidase C, that COI gene, is emerging as a standard barcode region for higher animals. In plants, some regions in genome considered as potential candidates for a barcode include MATK, tRNH, PSBA, RPOC1, RPOB, etc. Though at present there is no consensus on a universal barcode in plants. Now, the applications of molecular biological methods in plant taxonomy. Phylogenies based on RBCL sequences have been successfully obtained at the level of a family and higher taxonomic levels. In case of families like the Cornaceae, the Cupressaceae, the Ericaceae, the Geraniaceae, the Onagraceae and the Saxifragaceae, phylogenetic relationships have been inferred at inter- and intrageneric levels also. The morphology-based classifications of tribes and subtribes in case of Papilionaceae are mostly congruent with RBCL phylogeny. Non-coding regions of cpDNA have been found to be appropriate for working at lower taxonomic levels in genus Gentiana. Analysis of RFLPs and fasciolin indicated that a gene pool of wild Fasciolus vulgaris distributed between northern Mexico and northern Argentina consists of two major groups, Mesoamerican and Andean, and a third intermediate group found in northwestern South America. The classifications based on AFLP in case of Solanum were found to be in agreement with current taxonomic opinions. Unexpectedly, however, Solanum microdontum was associated with series Magistacroloba rather than with series Tuberosa, and Solanum demissum series Demissa and species of series Acola appeared closely affiliated. Similarly, use of single to low copy RFLP and RAPD data revealed paraphyletic nature of Solanum brevicoli complex. Separation of Juniperus into three sections, that's Caryocydrus, Juniperus and Sabina, has been supported by RAPD analysis. In this case, RAPD analysis was found to be taxonomically relevant at subgeneric level. RAPD analysis at the level of species in case of Allium also was in broad agreement with classical classifications, except that Allium royale was found to be closely related to Allium cepa in contrast to previous work. SSCP is used for rapid identification of Phytophthora ramorum, which is an important quarantine plant pathogen worldwide. The Pleurotus eryngi species complex was resolved using Isozyme and RAPD data analysis. Cytotaxonomic and enzyme electrophoretic studies have uncovered cryptic species in many fern lineages such as Adiantum, Botrychium and Pterochroma. Cryptic species were recognized in Asplenium nidus by Yatabe and Murakami in 2003 using RBCL sequence data. The most complete phylogenetic scheme of cyanobacteria, which is currently available with us, is based on 16S rRNA sequence analysis. With this useful role of molecular biology techniques in plant taxonomy, I conclude the present lecture and say thanks to you.